Imagine a vast library, not of books, but of instructions hidden inside every cell. This is the human genome. It's our personal book of life, written in a four-letter code, A, T, C, and G. Over 3 billion letters long, this code shapes everything from our eye color to our health. For centuries, its secrets were a mystery, but the ambition to read it grew with every scientific breakthrough. The Human Genome Project set out to read this code, creating a reference map for all humanity. This was not just science, it was a journey to the core of what makes us human. The Human Genome Project was a global effort, scientists from 6 nations united to decode our DNA. The genome's vastness required dividing the work, like assembling a massive jigsaw puzzle. Each team sequenced a section, sharing discoveries instantly online for all to use. This open access approach fueled rapid progress and ensured the knowledge belonged to everyone. Launched in 1990, the project was a bold, risky venture, but the vision of transforming medicine drove them forward. The result, a worldwide collaboration that changed science forever. Why map the genome? Because genes are the recipes for life, and even a tiny error can cause disease. By reading the full sequence, scientists could pinpoint genes linked to conditions like cystic fibrosis and cancer. This knowledge shifted medicine from treating symptoms to understanding root causes. The project opened the door to personalized medicine, treatments tailored to your unique DNA. It also allowed us to predict, prevent, and better understand complex diseases. The Human Genome Project was the first step toward a healthier future for all. The Human Genome Project united thousands of scientists, biologists, engineers, computer experts from around the world. Each country brought unique strengths: robotics from Japan, genetics from France, sequencing power from Germany, and more. Leaders like Francis Collins and John Sulston coordinated this global machine, overcoming political and technical hurdles. Behind the scenes, countless unsung heroes prepared samples, wrote software, and tackled ethical questions. This was more than a scientific project. It was a new model for international teamwork. Together, they built a genetic map and a legacy of collaboration. The genome is humanity's shared inheritance, so its exploration became a global responsibility. Major research centers each sequenced different chromosomes, working in harmony like an orchestra. Friendly competition and open sharing of breakthroughs accelerated progress for everyone. The project forged lasting international relationships and trained a new generation of global scientists. It's legacy proof that science can unite nations for the common good. The Human Genome Project showed that together we can solve humanity's greatest challenges. The project's most radical decision was to make all data public, posted online within 24 hours. This open access policy democratized research, letting anyone, anywhere, use the genome as a resource. It unleashed a wave of innovation, saving time and money for scientists worldwide. When a private company tried to patent the genome, the public team doubled down on openness. Their race wasn't for profit, but for the principle that the book of life should be free for all. This spirit of sharing became the project's defining legacy. Celera Genomics turned the Human Genome Project into a public spectacle, portrayed as David versus Goliath. On one side, the publicly funded consortium, sharing data freely. On the other, Celera, led by Craig Venter, aimed to charge for database access, raising ethical questions. The public consortium saw it as a battle for the soul of science, racing to keep the genome public. The pace accelerated incredibly, spurring innovation. The race captured global imagination and galvanized public support. In June 2000, a joint announcement declared a working draft of the human genome sequence. The result was a victory for humanity, ensuring free access to foundational data. Decoding the human genome was the culmination of decades of scientific discovery. The discovery of the DNA double helix in 1953 by Watson and Crick, building on work by Franklin and Wilkins, was the starting point. Reading the sequence of A, T, C, and G was a far greater challenge. In the 1970s, Frederick Sanger developed a revolutionary method to read DNA. Sanger's method became the cornerstone of the Human Genome Project. In the mid-1980s, the idea for a large-scale project began to take shape. Creating a complete reference map of the human genome would transform biology and medicine forever. In 1990, the Human Genome Project was officially launched. The first steps of this giant leap for biology had been taken. The project began by creating detailed genetic and physical maps of each chromosome. These maps would act as a scaffold for the sequencing work that would follow. The Human Genome Project required a robust strategy. Reading 3 billion letters was impossible with the technology of the time. The DNA inside our cells is incredibly long. Scientists broke it into smaller pieces for mapping and sequencing. The mapping phase created a table of contents, telling them where each piece of DNA belonged. The mapping process was twofold: genetic linkage and physical maps. The physical map specified the distance between landmarks. Once a physical map was complete, sequencing could begin. Each fragment was broken down further and fed into a sequencing machine. Computers stitched the small sequences back together, revealing the genetic code. The public Human Genome Project used the clone-by-clone approach. First, a human chromosome was broken into large, overlapping pieces. Each piece was inserted into bacteria to create perfect copies, or clones. This collection was called a clone library. Next, scientists figured out the correct order of these clones. Only then did sequencing begin, using a shotgun method to break each clone into tiny fragments. Because the origin of each fragment was known, computer assembly was simpler. This slower method prioritized accuracy, creating a reliable reference sequence. With DNA fragments prepared, the core task began: reading the sequence. Automated sequencers used an improved version of Frederick Sanger's method. Millions of copies were made for each DNA fragment, then used in a chemical reaction, creating new DNA strands that would stop growing whenever they encountered a specific letter. Each letter was tagged with a different colored fluorescent dye. Next, pieces were sorted by size using electrophoresis in a capillary. An electric current pulled fragments through the gel, sorting them by length. As fragments passed a detection point, a laser made the dye glow. A detector read the color, recording the corresponding letter. The data was sent to a computer. This automated process was a huge leap forward, essential for tackling the human genome. Years of mapping and sequencing led to a historic moment: the first draft of the human genome. On June 26, 2000, the world gathered for the announcement. President Bill Clinton declared the completion of the working draft. It was a globally celebrated milestone. This moment captured the public imagination, heralding a new age of genetics and medicine. Publication in Nature and Science was a landmark event, providing researchers a landscape of our genetic code. The work was not over. With the final sequence announced in 2003, it would be the project's lasting legacy. The Human Genome Project's immediate challenge was its unimaginable scale, accurately reading and recording 3 billion chemical letters. At the start, DNA sequencing technology was slow and expensive. To overcome this, the project became a catalyst for technological revolution, inventing entirely new tools. Scientists automated the process, developing robots and refining sequencing machines. Research centers transformed into massive sequencing factories, operating on an unprecedented scale. It required a new kind of scientist, expert in both genetics, process management, and quality control. Securing billions in public funding required immense political will, public support, and skilled communication. Beyond its size, the genome structure presented a challenge. Our DNA isn't a straightforward text. Large portions are incredibly repetitive. These junk DNA regions were a nightmare for sequencing. The shotgun sequencing approach relies on finding unique, overlapping sequences. In repetitive regions, every piece looked the same. It was like a jigsaw puzzle of a clear blue sky. The clone-by-clone strategy was helpful, providing a framework to bridge repetitive sections. Another challenge was the centromere, packed with repetitive DNA, difficult to sequence accurately. Telomeres, the chromosome ends, also presented a challenge due to their repetitive sequences. Overcoming these hurdles required persistence and ingenuity, showing that reading the book of life was about navigating its complex terrain. As sequencing factories operated, they created a tsunami of data. This data had to be stored, managed, and analyzed. The solution came from computing. The Human Genome Project spurred bioinformatics, merging biology, computer science, and statistics to analyze biological information. Computer scientists wrote new programs to assemble sequenced fragments back into chromosomes. Data storage requirements were immense. This information was stored in databases accessible to scientists worldwide. The next challenge was to make sense of it. Bioinformaticians developed tools for annotation. Without bioinformatics, the data would have remained unreadable. The Human Genome Project raised complex ethical challenges regarding privacy and discrimination. Project leaders addressed these issues by dedicating a portion of the budget to studying the ethical, legal, and social implications, ELSI. This program brought together experts to develop safeguards, research policy, and educate the public. Their work led to legislation like the Genetic Information Non-Discrimination Act, GINA. The project sparked debates about genetic testing, enhancement, and the future we want to create. ELSI fostered dialogue, ensuring the wise use of genetic knowledge. When the Human Genome was published in 2003, it was like opening a book for the very first time. Scientists dove into the 3 billion letters, expecting answers to biology's biggest questions. What they found was deeply surprising. Humans have only 20 to 25,000 genes, similar to a roundworm. Complexity comes from how genes are used. Vast regions between genes, once called junk DNA, regulate genes. Genes make up less than 2% of our genome. Scientists study the other 98% to understand how we work. We can now compare our blueprint to other species. We share 99% of our DNA with chimpanzees. These comparisons provide evidence for evolution. It's a story about our connection to all life on Earth. The Human Genome Project's impact on medicine has been significant. The completed sequence acts like a superpowered search engine for doctors and researchers, revolutionizing the diagnosis of genetic disorders. Conditions that were once complete medical mysteries can now be diagnosed with a simple blood test. The project also transformed our understanding of common diseases like cancer, heart disease, and diabetes. Perhaps the most significant change in medicine has been the development of precision oncology for cancer treatment. This personalized approach to cancer care is a direct legacy of the Human Genome Project. The Human Genome Project reinvented biology, ushering in an era of discovery-driven science and big data. Scientists could now analyze entire systems, transforming our understanding of life's complex machinery. This required a new kind of biologist, blurring lines between biology, computer science, and statistics. Bioinformatics became essential. The project also revolutionized DNA sequencing technology, making it faster and cheaper. This democratized genomics, spurring comparative genomics and providing a unifying framework for all of biology. It opened up the entire library of life. The Human Genome Project has profoundly impacted our understanding of ourselves and our past. Our DNA tells the story of our species' journey out of Africa and across the globe. By comparing genomes, scientists reconstruct ancient migration routes and map population expansion. The project scientifically disproves old ideas about race. We are remarkably similar. The genome proves we are one extended human family. This understanding has fueled ancestry testing, connecting individuals to their past. This has sparked public interest in genetics and our shared history, fostering a sense of global community. It has shown us our shared history as a single human species. It mapped the biological essence of our humanity. The Human Genome Project gave us the standard reference book for humanity, but the true future lies in reading our own individual copies. The cost of sequencing a human genome has fallen at a staggering rate. What once cost billions of dollars and took over a decade can now be done in a single day for a few $100s. This incredible accessibility is pushing us into the era of truly personalized medicine. Imagine a future where your doctor sequences your entire genome as part of a routine checkup. This information could then guide your healthcare for the rest of your life, creating a medical plan that is uniquely tailored to you. Another exciting frontier is pharmacogenomics, the study of how our genes affect our response to drugs. In the future, before prescribing a drug, a doctor will be able to check your genetic code to predict whether that drug will be effective and safe for you. As the cost continues to drop, it will become woven into the fabric of everyday healthcare for everyone. This is the direct and tangible future that the Human Genome Project has made possible for all of us. Reading the Book of Life was the first monumental step. The next, even more profound step, is learning how to edit it. Scientists have developed CRISPR, which functions like genetic scissors. It allows scientists to find a specific gene, cut the DNA, and replace it with a healthy copy. This incredible power opens possibilities once confined to imagination, especially in curing genetic diseases like sickle cell anemia and cystic fibrosis. Clinical trials show promising results, offering hope for a cure. Gene editing could also fight complex diseases, like engineering immune cells to destroy cancer. The potential applications are vast, but this power brings ethical responsibilities. The future depends on wisdom and public debate. The legacy of the Human Genome Project extends far beyond medicine. The ability to read and write DNA is transforming our world in countless ways. In agriculture, scientists are using this knowledge to develop crops that are more nutritious, more resistant to drought and pests, and have higher yields. This could be a crucial tool in feeding a growing global population in the face of climate change. By understanding the genes that control a plant's growth and resilience, we can create a more sustainable and secure food supply for the future. Genomic technologies are also revolutionizing our understanding of the planet's ecosystems. Scientists can now take a sample of soil or seawater and sequence all the DNA within it. This field, called metagenomics, reveals the vast hidden communities of microbes that live all around us. These microscopic organisms are the engines of our planet, driving nutrient cycles and maintaining the health of our environment. By understanding their genetic code, we can protect fragile ecosystems, clean up pollution using microbes, and discover new natural products from the world's incredible biodiversity. Furthermore, DNA is becoming the ultimate data storage medium. Scientists can translate any digital information, books, pictures, music, into a sequence of A's, T's, C's, and G's and store it in synthetic DNA. DNA is incredibly dense. A small test tube could hold all the data ever created by humanity. It's also remarkably stable, lasting for thousands of years. This technology points to a future where our archives could be stored not on fragile hard drives, but in the most reliable information molecule known to science. The world the Human Genome Project has opened up is one of incredible promise and possibility. The language of genetics is becoming central to human progress. The journey that began with reading our own book of life has given us tools to improve lives and become better stewards of our world. The future is written in the 3 billion letters of our genome, and we're only beginning to learn how to read it. The Human Genome Project stands as one of the great monuments of human exploration. It was not a journey to a distant continent or a new planet, but a voyage deep into the intricate, microscopic world that exists within each of us. The completion of this project was more than just a scientific milestone. It was a fundamental shift in our perception of ourselves. It provided humanity with its own instruction manual, a detailed blueprint that revealed the common genetic thread that binds us all together as a single species. The echo of this achievement resounds to this day, shaping the very foundations of modern biology and medicine in ways we are only just beginning to appreciate fully. The project's success was a testament to the power of human collaboration. It showed that when scientists from across the globe set aside competition and unite for a common cause, they can achieve the seemingly impossible. This spirit of open, shared discovery, where data was made freely available to all, created a resource that has enriched the entire world. It accelerated the pace of research in countless fields, empowering scientists everywhere to build upon a shared foundation of knowledge. This legacy of cooperation serves as a powerful model for how we might confront the other great challenges of our time, from climate change to global health crises. Looking back, the project was a gamble. It required immense investment, technological innovation on a massive scale, and the dedicated work of thousands of people over more than a decade. But the payoff has been immeasurable. It has given us a new vocabulary for understanding health and disease, moving us away from simply treating symptoms towards tackling the root genetic causes. Every new targeted cancer therapy, every diagnosis of a rare genetic disorder, and every step towards personalized medicine is a direct result of the foundational map that the Human Genome Project so painstakingly created for the benefit of all humankind. The story of the Human Genome Project is ultimately a story of curiosity and hope. It was driven by our innate desire to understand who we are and by the hope that this knowledge could be used to alleviate suffering and improve human lives. It was an audacious promise, and one that has been delivered upon time and time again. The completion of the sequence was not an endpoint, but a glorious beginning. It opened a new door of perception, inviting us into a world of incredible complexity and beauty, and setting the stage for the next chapter in our species' long and remarkable journey of discovery. With this profound new knowledge comes an equally profound responsibility. To hold the map of our own genetic code is to hold a power that previous generations could only dream of. The ability to read, and now even edit, the Book of Life brings with it a host of complex ethical questions that we, as a society, must navigate with wisdom and care. This knowledge can be used to heal and to help, but it also has the potential to be misused. The challenge of our time is to ensure that the fruits of this incredible scientific revolution are used to foster equality and benefit all of humanity, not to create new divisions or forms of discrimination. The conversations started by the Ethical, Legal, and Social Implications program are more important now than ever before. As genetic testing becomes more common, we must continue to protect individual privacy with robust laws and secure technologies. We must ensure that a person's genetic information cannot be used against them by employers or insurers. It is vital that access to the benefits of genomic medicine is equitable, available not just to the wealthy, but to everyone, regardless of where they live or what their circumstances may be. The promise of the genome must be a promise for every single person. The advent of gene editing technologies like CRISPR presents perhaps our greatest ethical test. We stand at a crossroads. One path leads to incredible cures for devastating diseases, correcting genetic errors in patients to relieve suffering. The other, more perilous path leads towards the possibility of heritable genetic modifications, making changes to the human germline that would be passed down through generations. This latter path raises the specter of designer babies and could fundamentally alter the human gene pool. It requires a deep and global consensus on where we should draw the line, guided by caution, humility, and a shared sense of our common humanity. Ultimately, scientific knowledge itself is neutral. It is a tool. The morality lies in how we choose to use it. The legacy of the Human Genome Project is therefore not just a scientific one, but a social one. It calls upon us to be thoughtful guardians of this powerful information. It requires ongoing dialogue between scientists, policymakers, and the public to build a future where genetic knowledge is a force for good. Our ability to act with wisdom and foresight will determine whether this new chapter in human history is one of triumph and healing for all. The Human Genome Project did not provide all the answers. Instead, it revealed a landscape of inquiry that is vastly larger and more complex than we ever imagined. It was like reaching the summit of a great mountain, only to see countless other peaks stretching out towards the horizon. We have the sequence of letters, but we are still in the very early stages of understanding its intricate language. We have identified the genes, but we have yet to uncover the full function of most of them, or to decipher the complex symphony of regulatory switches that control their expression. The Book of Life is open, but its most profound secrets are still waiting to be read. The future of genetics is an endless frontier, full of promise and wonder. We will continue to unravel the complex genetic risk factors for common diseases, leading to ever more precise forms of preventative medicine. We will explore the dark matter of the genome, the 98% that does not code for proteins, and discover the crucial roles it plays in health and disease. And we will begin to understand how our genes interact with our environment, our diet, and our lifestyle in the intricate dance that makes each of us who we are. Each new discovery will open up new avenues of research and new possibilities for improving human well-being. This journey of discovery will not be limited to human health. The tools and knowledge born from the Human Genome Project will continue to help us address some of the world's most pressing challenges. From developing sustainable agriculture to protecting biodiversity and understanding the vast microbial world that supports our planet, the language of DNA is becoming a universal key. It allows us to read the stories of all living things, to understand our place within the web of life, and to become better stewards of our precious home. The future is one where biology and technology work hand in hand to create a healthier and more sustainable world. The quest to understand the human genome is, in the end, a reflection of the human spirit itself. It is a story of our insatiable curiosity, our drive to explore the unknown, and our hope for a better future. The project has given us a new and humbling perspective on ourselves, revealing our shared history, our deep connection to all life, and the astonishing complexity packed within every cell. The journey is far from over. The frontier of the genome is vast and full of wonder, and the exploration of its mysteries will continue to captivate and benefit humanity for generations to come.